Lesson 23, Neuronal Function. In this lesson, we are going to discuss the electrophysiology of nerve function, different types of channels, resting membrane potential, local and action potential, and how are the action potentials generated and propagated. Now, uh, starting with the ion channels. In nerves, uh, proteins are required for all ions to pass through the plasma membrane. There are two main classes, leak channels and ion channels. Leak channels, as the word suggests, it will be always open. Ions move down concentration gradient into or out of the cell. You see in this first part of the picture on the top, where the ions are moving freely in and out due to the um, concentration gradient from higher to lower concentration. It is always open. For the second type of channels, the ligand gated channel, there are three gated channels: ligand gated, voltage gated, and mechanically gated channels. These gated channels are closed at rest and open in response to certain stimuli. When it opens in response to chemicals that bind to the channel, we call it ligand gated. In the second part, you see that the gate is closed first, but the moment that green ball, the ligand, is attached to the gate, which is the chemical messenger, that moment the gate opened for those chemicals to move in and out. Next channel is the voltage gated, open or close in response to changes in voltage across the membrane. In this third picture that you see here, initially it is closed and the voltage was positive outside, negative inside. The moment it changed, that negative outside, positive inside, the gate opens. Fourth and last type of channel will be mechanically gated channels, which opens and closes in response to stimulation, such as pressure, vibration, or stretch. You see this picture, in the lower part, that the gate is closed first. The moment there's something touched it, which, as I mentioned, it can be pressure, vibration, or a stretch, those gates will open. Now, most cells, extracellular sodium is higher than in sodium, in cytosol. Okay, the sodium is more outside the cell. This is what it means than inside. On the other hand, potassium will be more inside than outside. So keep that in mind. In most cells, sodium is higher in outside and potassium is higher inside. And this gradient, why is that different? Because the gradient is maintained by sodium potassium pump. And that maintains the resting membrane potential. The potential forms from separation of charges across membrane, and the potential is called voltage, a type of electrical gradient across the cell membrane. It is the membrane potential, or Vm. We express this with Vm, membrane potential. The voltage of the inner membrane is negative relative to outer membrane because you know the sodium potassium pump, it moves 3 sodium out and gets let 2 potassium to get in. So it will be negative, inner membrane would be negative, and outer side will be positive. Negative voltage is present when cell is at rest, and it is called resting membrane potential, RMP. Cell in this state is polarized. Okay, so if we take a small part of the axon, this is what we see, the difference between the voltage, which is negative 70 millivolt. Okay, now the generation of RMP. The ion concentration favors diffusion of potassium out of the cell and sodium into the cell, because we learned earlier that sodium is higher outside, potassium is higher inside. The potassium leak channels diffuse more easily than does sodium because of the concentration gradient. So this is, they try with the leak channels, they try to move in and out. Diffusion of ions is determined by concentration gradient and electrical gradient. Combined forces is called electrochemical gradient. Right? So one is the electrical, other one is the chemical concentration. Together we call it electrochemical gradient. So this picture will clarify the things we just mentioned in last few slides. Look at this channel, the leak channel. This is the potassium leak channel. You see the blue one at the middle. Now, the concentration of potassium is higher inside the cell, which is cytosol, right? So this concentration gradient favors the movement of potassium to the extracellular fluid. So it tries to go outside. That makes sense. The electrical gradient favors the movement of potassium to the cytosol, because you see the gradient, it's positive outside, negative inside. So potassium is positive. That favors, this gradient favors the potassium to go inside the cell. Okay, so two forces you see here. One, according to the concentration gradient, it tries to go outside, but due to the electrical gradient, it tries to go inside. Ultimately, the concentration gradient is slightly stronger, so a small force favors the movement of potassium into the extracellular fluid. Now, we know what is resting membrane potential and how is that working. Let's talk about the action potential. When a sodium channel, gated sodium channel opens, it allows sodium down their electrochemical gradient and rush into the cell. All right, so sodium will enter inside the cell, resulting in cells gaining positive charges, making the VM more positive. Okay, so inside the cell, it was negative, but when the sodium enters, it goes to positive. And this is what happens. You see here, from negative 70, it is become going towards the positive side, negative 60. Okay, so this is depolarization. The gaining of positive charges makes the inside of the cell less negative, and this is called depolarization. Now, when the cell returns back to its resting membrane potential as the sodium channels close, that will be repolarization. So it's the opposite thing. Sodium channel opens, sodium comes in, then it is the depolarization. And when the channels close, then potassium is actually going outside because of the leak, and that will become negative again. That will be depolarization. So that was the channel we talked about. That's the sodium and potassium. But when the voltage gated sodium channel open, okay, and the membrane potential may become more negative than resting membrane potential, this will be hyperpolarization. So three things: depolarization, repolarization, and hyperpolarization. In hyperpolarization, loss of the positive charges makes the inside of the cell more negative, as you see here. It's going downwards and causing hyperpolarization. So if I summarize these three events, this is what you see. Um, that the depolarization, that's number one is C, when opening of the gated sodium channels allows sodium to enter. Then step two, repolarization, that is B, when a cell returns back to its resting membrane potential as sodium channels close. And then hyperpolarization, three, will be the A, that is voltage gated potassium or anion channels open, membrane potential may become more negative than resting membrane potential. Graded potential or local potential. Small local change in the membrane potential of a dendrite or cell body. It may be depolarizing or hyperpolarizing. These are local or graded potential. Okay. What about action potential? Uniform, rapid, and all or none depolarization and repolarization of the membrane potential. In nervous system, action potentials are generated by a trigger zone and sent along the axon to another neuron, muscle fiber, or gland. Right? So, what is action potential? It has the whole mechanism we'll discuss, but first thing first, that this is the way the nerves can start their trigger zone and send this along the axon to another destination. Let's talk about the events of an action potential. Neuronal action potential have three general phases. Each phase occurs because of opening and closing of ion channels. We just want to start here. And the first picture on the top, you see here the beginning of the axon. A local potential depolarizes the axolemma of the trigger zone to threshold. So that's the trigger zone at the beginning of the axon here. Okay. And this is the when this local potential starts here, that will open up the voltage gated sodium channels. So you see the pink color sodium channel, they will open, sodium will enter, and the axon will, this part will be depolarized. Okay. So we started with the local potential, then second thing, the depolarization happens because the sodium channel is open and the charge is going very high. The voltage is from negative 70 to plus positive 30. What happens next? Sodium channels inactivates. It closes. And the voltage gated potassium channels will activate. So sodium stop entering and potassium exit the axon. Repolarization begins. Okay. In next step, step 4, sodium channels return to the resting state. Okay. And the repolarization continues. Potassium is moving out, and that brings the voltage lower to the close to negative 70. But that moment, the axolemma may hyperpolarize before potassium channels return to the resting state. After this, the axolemma returns to the resting membrane potential. So this is the point where we see is the hyperpolarization. So the bottom line is depolarization followed by repolarization and hyperpolarization. Neurons are limited to how often they can fire action potential. There is a limitation, and that is called refractory period. It is the time the membrane cannot be stimulated to fire action potential. The another one, it is refractory to that. Refractory means when it has started to fire. At that moment, if there is a second stimulus, it may not work on that. So it may not be able to be stimulated for the second stimulus. That is the refractory period. There are two types of refractory period, absolute and relative. You see this picture on the right side first, right? We know what is the resting membrane potential, -70. Then we have this local potential, it brings it to -50, then depolarization, followed by repolarization, hyperpolarization. So in this part, you see the absolute refractory period part, this area, there will be no additional stimulation, no matter how strong, can produce an additional action potential. So even if we stimulate the nerve with a stronger things, stronger stimulus, this is not going to work during this time. The absolute refractory period, when the depolarization and repolarization is happening, at that time there will be no stimulus, no additional stimulation will make this an additional action potential. But after that, when the hyperpolarization stage, only a stronger stimulus than normal will produce this action potential. This is called relative refractory period. Okay. Now, the local versus an action potential, if we compare it, in the local action potential, local potential, it is graded. Action potential is not graded, it's uniform. Local one is reversible, the action one is irreversible. Local potential is decremental, and action potential is non-decremental. Action potential must be propagated along the axon. Okay? And how is that happening? This is the thing we are discussing in next few slides, the propagation of action potential. Movement creates a flow of charged particles or current. Action potentials are self-propagating. They occur in one direction, and propagation forms the nerve impulse. So let's talk about the step one. First step, we consider this beginning of the axon. And you see at one side of the axon, the axolemma, what is happening. That's the trigger zone, right? Okay. At this point, the trigger zone, the depolarized membrane is here. Okay. When it is depolarized, we know the inside the cytosol, it becomes positive. And because the sodium is more inside that area, it enters there. Now, and that is the initial point, the trigger zone. Okay. As the sodium channels activate in the second area, the action potential is triggered and it spreads down the axon. So you see it is moving from left to right. At one point, it started with the trigger zone. It opened up the sodium channels in the next area, and the first area become resting membrane potential. The second one is having action potential. After that, it will keep going to the third zone and then fourth zone. So it moves, it propagates. All right. So this is how the propagation of action potential happens. This is speed of conduction of the action potential is determined by diameter of axon, diameter and the presence or absence of myelin sheath. Diameter of axon, the larger axon have lower resistance to conduction. So it will move faster. and current flows through them more easily if the diameter of axon is large. The presence of myelin sheath causes saltatory conduction, and that makes it faster. Flow of current is more efficient in a myelinated axon and significantly faster. And what is saltatory conduction? We need to just have a look at this. Look at the picture on this slide. You see the saltatory conduction, the depolarization membrane happens in the first action potential, then the second part, then third part. So it goes like a section, one at a time, and that makes it faster. Other than if you compare it with the absence of myelin sheath or unmyelinated one. In that case, the continuous conduction occurs like this one. You see here that goes slowly. Okay, slowly it is going on. But if you compare it with the myelinated one, that one happens segment at a time, one segment at a time. It's like a bunny hopping animal or like a kangaroo. It's like a jumping and jumping like that. Three main classes of um, axons according to the conduction speed: A, B, and C. Three types of fibers. In type A axon fiber, it is the largest one. Type B will be intermediate. Type C will be smallest. So larger fibers will be myelinated, all of them. Type B most, but type C none. And larger fiber myelinated, definitely it will have the maximum velocity. Action potential will move faster, like 120 meter per second. If you compare it with the type C fiber, which is zero point 0.5 to 2 meter per second. So it is almost like 100 times or 50 to 100 times faster. Examples of this type A fiber are those sensory axons from joints and muscles, the motor axons of the skeletal muscles. This is why we can move our skeletal muscles so fast. Within a fraction of a second, we can do that because the nerve carrying this impulse to the muscle is the type A fiber. In comparison to the nerves in the autonomic nervous system or some sensory axons of the that transmit pain, temperature, or certain pressure sensation, it takes a while, some time, because they have type C fiber. So this is our last slide for this lecture. What we see here, just before we finish, to summarize, you see the local potential usually generated in the neuron's dendrite, uh, accumulate and reach the trigger zone of the axon. Okay, this is the trigger zone you see here. And then this action potential, the trigger zone depolarizes to threshold and generates an action potential that will move on. And at the end, the action potential propagation will take place down the axon and up to the axon terminals. So that is all about this lesson.